and I have covered vitamins also in details, which are always, I say, are most important. Vitamins, बहुत important है यार, vitamins तो हमेशा ही important होता है. ठीक है, यहाँ पे at this session we will be covering the genetics part, right, the molecular part of biochemistry. Fine. And one thing again I want to tell you is that that I was telling that NEET PG this time there were very less questions from this molecular section. So this time there may be lot of questions from these topics, maybe, right? Okay, the exams are unpredictable, but yes, I feel like INI-CET is a little bit predictable because AIMS and all these exams, they always repeat the questions at least almost 50%, I feel. Fine, so let's start without wasting time, right? Okay, and as the tagline of our institute is like padhayenge utna zaruri jitna, right? We'll be covering only the key points which are super important for the exams, right, in the form of MCQs. Let's start. So question number 1. So question number 1 is not correct regarding the human DNA. Okay, so guys, human DNA, right? Human DNA, normally the human DNA which is found in our system is the B type of DNA, right? That is normally the B type of DNA physiologically. So that was discovered by Watson and Crick, and using the technology that is X-ray crystallography, right? So what was the technique they used? X-ray crystallography. So now, first of all, it is left-handed double helix. Is it correct? No, it is a right-handed helix. So this is our answer, right? Option A. It is negatively charged, yes, because of the presence of phosphate, right? Because of the presence of phosphate. GC-rich DNA has a high melting temperature. Because that is again a question which has been asked previously, right? So there is a term that is called as DNA denaturation, right? Okay, so there is a term that's called as DNA denaturation, and DNA denaturation occurs at a high temperature of around 90 degrees Celsius, right? So the two strands of the DNA separates. Fine. So those DNA which has G and C, right, obviously they have the 3 hydrogen bonds, so that is the DNA which is less prone to denature easily, right? So that is one point. So guys, these are hydrogen bonds, okay, and there are two things which prevents the DNA denaturation easily. So what are these bonds? Okay, hydrogen, okay, one is called as hydrogen bonds, right, and the base stacking forces. What is the base stacking forces? Like AT is stacked over I mean, like there is GC, then there is AT, then GC, something like that, right? So these are called as base stacking forces. So these are the two forces, right, which hold the DNA tightly. Okay, so the DNA denaturation. So this is the point which has been recently asked in exams. So I'm repeating, I am covering only the points which are asked in exams or has been part of the MCQ as an option, right? Okay, so GC rich DNA. So how else you can increase the denaturation of the DNA? Guys, you can increase the denaturation can be increased by increasing the pH of the solution and decreasing the salt concentration. Guys, why? Because salt concentration conceals the phosphates, right, and prevents the repulsion between the negative charges. So salt concentration increase in the pH, breaking the hydrogen bonds, that is by heating. Okay, so that is heat. So these are the 3 factors which can denature the DNA also. And guys, DNA, after denaturing the DNA absorbs light at what particular wavelength? 260 nanometers. So increased absorbance, single stranded DNA, right? Single stranded DNA has increased absorbance at 260 nanometers, right? So remember that point also. Fine. Now option D, binds with histones. So guys, histones are the positively charged proteins, and histones are mostly rich in lysine plus arginine, right? And histone octamer, one more question I want to add here. Histone octamer, when we make a beads on the string appearance, right? When we make a beads on the string appearance, these are the octamers. Which is the histone which is not found in the octamers? Answer is H1, right? So histone has a lysine plus arginine, right? And there is no H1 in the histone octamer, right? So this is the octamer, but H1 is present somewhere in the linker DNA, right? Is linker DNA. That is again a question, that which of the following histone is not found in histone octamer. So answer is H1. Clear? H1. Okay, guys, fine. So that's it. This much you should know. Fine. And don't forget to revise Chargaff's rule, right? Basic thing I'm not covering here, but you should know. I'll keep on telling you, so keep your notebooks ready, right? Okay, I'll keep on telling you few things, few I will write. Okay, लिखते रहो यार, है ना, मतलब पता होना चाहिए, जैसे चारगाफ रूल आना ही चाहिए, must know चारगाफ rule. Okay, next is moving to the next question. Okay, so which of the following is correct sequence of enzymes required for DNA replication, right, or the DNA formation? So what are the correct sequences? See guys, first enzyme is always helicase. You know, helicase starts breaking the DNA and make a replication fork, right? Second enzyme can be topoisomerase because it keeps on releasing the supercoiling. Okay, so first enzyme should be a DNA helicase, right? Always. So I can rule out the option B and option D already. Okay, so after DNA helicase, the RNA primase is mentioned here, right? And in the C option, again RNA primase is so that is correct. DNA polymerase 3, then 1 and ligase. So guys, A is the answer, because in the C we have DNA polymerase 1 coming before the third. No, right? Third comes first in the prokaryotes. This is what we are talking about, the prokaryotic replication. Okay, so the third comes, yes, Dr. Shubha. Fine. So the third comes first, right? So guys, DNA polymerase 3 is something which synthesize both the leading and the lagging strand of DNA, right? Okay, so that is what is activity 5' to 3' polymerase. Fine. DNA polymerase 1, what is the activity, guys? It is the activity which is, you know, primer removal. So if I have to compare DNA polymerase 1 and 3, right, old MCQ, so you know guys, one has one activity that is called as 5' to 3' exonuclease activity, you know, and that is for the primer removal. Okay, that is for primer removal, and this is not found in the polymerase 3. So don't forget this activity. Okay, so this is the activity which is uniquely found in 1 for the RNA primer removal. Okay, that is the point that is. And guys, there is a term that is called as clinofragment. Okay, that is again one of the favorite question. Clinofragment. Clinofragment, what is clinofragment? Clinofragment is the DNA polymerase 1 without this activity, right? Okay, so this activity is not present. Fine. Which activity? 5' to 3' exonuclease is not present. So that is called as clino. And there is one more term that is called as exoclino, right? As clinofragment was initially used for in the PCR, right? Because it is not removing the primer. And during the early phases of PCR, we wanted some enzyme which doesn't remove the primers, because in PCR we don't do that. But in the cell we have to do that, right? So that is the term clinofragment. Fine, guys. Okay. So let's move to our next point. Fine. So guys, in case if the option changes, so topoisomerase will come after the helicase. You know, if the option is given in such a way that helicase then topoisomerase, so that is correct. And then they give the polymerase, that is RNA primase, and then the third, then first, and then ligase in the end. Okay. So topoisomerase, though it is working simultaneously with helicase, but initially the helicase starts the process. That's it, guys. Okay, nothing more than that you should know. So let's move to the question number 3. Again, very important. Question number 3. So Bloom syndrome. Bloom syndrome is a disorder of defective. So answer is defective helicase. Right? Helicase is the answer. Here. So in So guys, helicase, there are three disorders that you should know of the defective helicase. So first is definitely the Bloom syndrome. Okay, these are the disorders, which has a genomic instability, increased risk of cancers, and the faster aging or the premature aging, right? Second is called as the Werner syndrome, that is also called as adult progeria, right? And the third is called as the Rothmund-Thomson syndrome, right? Rothmund-Thomson syndrome. So just the names you should know, nothing more than that, right? Necessary thing. Okay, guys, looking at the other option, let's take option C. So polymerase, there is no disorder as such. Topoisomerase, there is no disorder as such. Though we have drugs like tropoticon and irinotecan, which is anti-cancer drugs, which are inhibitors of topoisomerase. Right. Now, moving to the next point, that is telomerase. As telomerase is very important. Okay, so some must know points on telomerase. Just I am revising. Telomerase is the enzyme that is also called as the reverse transcriptase. Okay, reverse transcriptase, don't forget this line, and that is RNA dependent DNA polymerase. Okay, RNA dependent DNA polymerase, right? D for dependent. That is the telomerase enzyme. And guys, we are talking about the disorder now. So defective telomerase, though never asked, okay, defective telomerase leads to a disorder where the bone marrow fails, and that is called as dyskeratosis, right? Congenita. Okay, at least you should know this name. And it has a triad of bone marrow failure and the nail dystrophy and the reticulated skin. Bone marrow failure, second is nail dystrophy, and third is reticulated skin. Okay, so that is one more point. Fine. Now, there's one more point. Telomerase is something, which is the cause of our aging, right? So loss of the end of chromosomes is a cause of aging, I mean to say, and telomerase can prevent that. So where is the telomerase found? Telomerase is found in certain cells like the germinal epithelium and the stem cells, and the cancer cells activate the gene of telomerase. That again we all know. Fine, the gene is activated by the cancer cells. Yeah, and guys, there is a term that is called as Hayflick limit that has been questioned. Hayflick limit. Okay, so what is Hayflick limit? That a normal cell can divide only for 40 to 60 times, and then apoptosis happens. Why? Because of decrease in the length of telomeres, the somatic cells, right? What only can divide for, so that is a limit of the cell division, 40 to 60 times. Yeah, guys. So that is all about this thing that I want to take here. Fine. Yes. Good. Fine. Great, guys. Great. Fine. So let's move to the next question. Right. We are covering the most important points. Okay. Nothing more than that. The last few days are remaining. Okay. Oh, I'm sorry. Like not. I should not say 10 to 15. Definitely, every day counts for you. Right. So we have few days left. So let's start with the question number 4. Guys, I just want to tell you one more thing, that during the exams, don't feel the aggression to do all the MCQs, right? At 200 MCQs and I want to do all of them. Attempt only around 170 to 180, not more than that, because that is one of the biggest cause of failure, the negative marking, right? Okay, so I suggest you're going for the exams, don't. Fine. Okay, so let's talk about the question number 4. So as Human Genome Project. Human Genome Project was something that started in 1990, and it was set to be completed in 2003. Right? So it didn't take 2 decades. Like one of the option was like that in one of the exam. So it took only 1 decade plus few more years. Fine. Now in the Human Genome Project, it was found that the double-stranded DNA has 3 billion base pairs. Okay, 3 into 10 raised to power 9, right? That is 3 billion base pairs. Fine. So obviously the haploid genome has 3 million. That is a wrong statement, right? So what is false regarding the human genome? Now let's talk about more points on the human genome. So guys, this is a project which is not covered in PSM, I mean to say, right? This is a project that was started in 1990 with the help of a few countries like Japan and US and a few more, right? So 3 billion base pairs are there. Okay, fine. Sonu sir will be the next. You're getting a point? Now guys, what is the goal of the human genome project? So what is the human genome project? Fine. So goal is to sequence the DNA, one thing, and localize the genes on chromosomes, right? So that is the goal. How it was done? Few more points that we should know, right? So guys, first of all, the DNA was isolated, then it was fragmented. So we took the DNA isolation, then DNA was fragmented or cut with the help of restriction enzyme. So DNA is fragmented, fine, and then guys, the small DNA fragments are ligated to, right? So DNA is ligated to the vectors. Guys, vector is something which carries the DNA, which transfers the DNA from one organism to the other organism, we can say. So ligated to vectors, right? So guys, what are the vectors used in the process of human genome project? That you should know. So they were bacterial. Anybody? What was the vectors used? Bacterial artificial chromosomes and yeast artificial chromosomes. I'll talk about them later again in one question, right? So these are the vectors. So guys, highlight this. These are the vectors. What can be important in the next exam MCQ. Then guys, after ligation, that is cloning, or they were copies were made, right? So what is the purpose of cloning the DNA into the vector? For example, this bacterial artificial chromosome. So we made copies, right? And these copies were sequenced, right? So what was the process of sequencing? Sanger's sequencing. So guys, again highlight this point. Sanger's sequencing was used, and these are the two types of artificial chromosomes are used. So guys, what are the findings of human genome project? Okay, that you should know, that you must know, I mean to say, because this question has been already asked for the first time in INI-CET. It has never been asked previously, so can be repeated. Fine. So what are the findings of human genome project? Few are already in our they are given in our MCQ, right? So what are the findings? Guys, the findings were that there are 30,000 genes, right? And that makes 3 billion base pair of total, right? Double stranded DNA. 30,000 genes are there. Out of this, chromosome 1 has the maximum genes, right? Chromosome Y has minimum genes, that is just 231. Okay, 231. Fine. So chromosome 1. Then guys, X chromosome has the largest gene. Okay, so that has the largest gene, that is of dystrophin. Okay, dystrophin, that is 2.4 megabases. Okay, or 2.4 million bases, right? So dystrophin gene. And Y chromosome, it has the smallest gene, that is called as SRY. Okay, that is the smallest gene. So that is also called as the testicular differentiation factor, and that is just 14 bases. Guys, that is just 14 bases. Clear? So that is again very, very important. That is Y chromosome. Fine. Then what else? Guys, average gene size is 3000 base pair. that is the average gene size, that you should know. Then one more point that we should know is given in our option, that is less than 2% of human genome code for proteins, or currently we say less than 1.5% actually, right? Important MCQ. Fine. So guys, these are the few important findings of the human genome. And centromere, right, and the telomere. So guys, they have the repetitive DNA, that is also called as the junk DNA. Fine. Junk DNA. So that is also the important point. Okay, fine. And guys, last point, that is 1.4 million single nucleotide polymorphisms are also present, that I can add here. Okay, that 1.4 million single nucleotide polymorphisms. What is a single nucleotide polymorphism? The difference in the DNA because of just one nucleotide. That is called as SNP. Okay, what is the difference between SNP and point mutation? SNP is present less than 1% in the population, and SNP is more than 1% in the population, and mutation is less than 1% in the. So that is how we can differentiate the point mutation from the single nucleotide polymorphism. So guys, Human Genome Project, I have covered everything that you should know, right? So moving to our next point, which of the following vitamin deficiency will prevent the thymine insertion in the DNA? Okay, thymine insertion in the DNA. So guys, the answer for this question is already highlighted. Answer is C, folate. Okay, folate is the answer. Why? Because to make the T from the uracil, right guys, DUMP to the DTMP, that is thymidine monophosphate, what we need is the enzyme that is called as the thymidylate synthase. Okay, and guys, what is the donor of the methyl group? That is called as methylene tetrahydrofolate. Okay, methylene tetrahydrofolate, which converts into dihydrofolate. So that is the donor of the methyl group. That is why folate is answer. Guys, B12 can also be the answer, because the active form of folic acid, or to release the folic acid from its storage form, that is a methylated tetrahydrofolate, we need B12. You remember in which step? That is in the during the methionine synthesis, right, from the homocysteine. We also need B12. That is why the B12 deficiency leads to folate trap. Okay, fine guys, that is a folate is the answer here. Very simple, right? Nothing much about this question. So question number 5. Right guys, now, so remember which vitamin deficiency causes folate trap? B12. Okay, fine. Guys, okay, so moving to our next question. Question number 6. Dideoxynucleotides are used in Sanger's techniques. Why? Right? Why? Because answer is given. It doesn't allow the further elongation of DNA. Right? Answer is A. It doesn't allow the further elongation of the DNA. Here. So rest of the options we can ignore. Fine. They can be made fluorescent and others, right? So guys, what is Sanger's technique? Sanger's technique is used for DNA sequencing. Okay, and this is based on the principle of the chain termination using dideoxynucleotides. Okay, what is the principle? This is chain termination using the dideoxynucleotides, right? Now guys, we all know one thing, that during the DNA replication we use the nucleotides which are made of pentose sugar, phosphate, and the B for base, right? And it has a OH group, right, which is present on the 3' but not on the 2' fine. So that is the 1', 2', 3', 4', and 5'. The OH group is present on 3', right guys? But if you remove this OH group, or O, this is called as a dideoxynucleotide. So guys, why this OH is important? To add the next nucleotide during the replication process. Okay, this is a B for base again, H and OH. So guys, this phosphate and this OH group, that is called as a phosphodiester bond, and how the DNA is being replicated, right? Or how the DNA is being synthesized. Yeah, so that is why if it is if this OH is not present, the phosphodiester bond cannot be made, because that is why this OH is very, very important. So dideoxy means both the OH are missing from second and third. Okay guys, fine. So hope you understood. That is called as Sanger's technique. Okay, it doesn't allow the further elongation of DNA. Okay guys, so moving to our next point, next question. Okay, so look at this question. A 7-year-old girl develops blister on exposure to sunlight. Irregular dark spots, right, on the skin are also found, right? He is very likely to have, she is very likely to have defect in which of the following repair mechanism? So guys, this is a case where the freckles develop because of exposure to the sunlight, and that's a case of, what is the case? Anyone watching me right now? Anyone answer? What is this case? This is a case of xeroderma pigmentosum, right? This is xeroderma pigmentosum. Xero means dryness, pigmentosum means pigment, freckles that develops here, right? It's very common in countries like India also, right? So nucleotide excision repair is defective here. So guys, what is the purpose of nucleotide excision repair? Okay, so nucleotide excision repair, right? So first of all, it removes the UV induced T dimers or thymidine dimers. So first thing that you must know is T dimers, right? So what does the UV rays causes to the skin? They make thymidine dimers, right? Or the pyrimidine dimers. Yeah, yes, good. So T dimers, fine. So they remove the UV induced T dimers, first thing. Then this is repaired by nucleotide excision repair. So what is the name of disease? Xeroderma pigmentosum, with increased very high risk of basal cell carcinoma and squamous cell carcinoma, right? Okay, in this image also we can see that, though it is not very clear, but this is the common site of what? The BCC, right? The most common site of BCC. Fine. Then guys, we have mismatch repair. So guys, what does a mismatch means? When the A bind with the C or the A bind with the G, for example, right? That is called as a mismatch repair. So what is the name of disorder? That is called as hereditary non-polyposis colon cancer, right? Hereditary non-polyposis colon cancer. Then we have base excision repair. Guys, what does a base excision repair do? That, this is the repair which removes the the wrong base, right? This will remove the wrong base. Fine, wrong base is removed. Clear? So wrong bases are removed, like for example, when the adenine is bound with uracil, right? Adenine bound with the uracil, for example, so you have to remove the U. Okay, and guys, this need the enzyme that is called as glycosylase. So I'm just adding those points which are part of or has been asked in exams previously. Glycosylase, right, which removes the uracil. Fine guys, so all of them are very, very important questions, right? Okay, so base excision repair, and there's one more thing, that there is defective non-homologous end joining, and this causes SCID, right? The autosomal recessive type of SCID, one of the common cause of that. So non-homologous end joining defective, again very important question, defective NHEJ. Now guys, can you answer this question? Or first of all, let me show you one point here. Okay, identify the genetic disorder and underlying mechanism. Can you answer this? This was the image given in one of the question, the INI-CET exam itself. Because this is what? This is called as a tiger tail appearance of the hair shaft, right? and that is seen in a condition that is called as, anyone? So the answer will be, that is called as something that is called as the trichothiodystrophy, right? This is called as trichothiodystrophy. And guys, trichothiodystrophy is something which is also called as Pibidis syndrome. Okay, Pibidis syndrome. So guys, this is again due to the defective nucleotide excision repair, right? Defective nucleotide excision. Okay. Right. What is Pibidis? Where the P stands for photosensitivity, I for the intellectual disabilities, B for brittle hairs, as you can see in the picture, brittle hairs, sulfur deficient brittle hairs, I for ichthyosis, right? D for the decreased fertility, and S for the short stature, right? So these are wherein it's not Mankey's, right? In the Mankey's there is copper deficiency, and there is the hair pattern which looks like the kinked hairs, kinky hairs. These are not kinky hairs. These are just brittle hairs, rough brittle hairs, right? Okay. In the kinky Mankey's disease, the hairs are folded, right, like this. Okay, and there is a pili torti, tortuous hairs, right? Pili torti is there. Okay, so that's your point. Okay, fine. So guys, this is again a disease of what? Nucleotide excision repair, that is trichothiodystrophy. Now, double strand break repair introduced by the Cas9 endonuclease are repaired by. So guys, this question has been asked several times, right? Okay. So Cas9. So Cas9 means we are talking about here CRISPR Cas9. So which was the Nobel Prize in 21. Okay. The discovery of CRISPR Cas9. Fine. Though it is already in our textbooks. It was discovered almost 10 years back, but the prize was given in 21. Okay, announced in 20. So non-homologous and joining is the answer, right? The question is double strand break repair. So guys, when we talk about double strand break repair, okay, so when the two strands of the DNA breaks, right, so they are repaired by non-homologous and joining. That means just join the two ends, whatever the fragment is lost, that is not replaced. So this is error prone. And the other is homologous or homology directed repair, that is HDR. Option B is homology directed repair. So homologous repair, right guys? Okay, so non-homologous and joining and homologous repair. Fine. So NHEJ is the most commonly utilized process to join these broken ends. So these are the broken ends, guys. Let's say double stranded DNA. So they will be just fused. Okay, they will be just fused using some proteins and some ligase enzymes. Yeah. So guys, if it is error prone, fine, because we are not replacing the deleted strand, right? Then why it is done commonly? Why we are not doing homologous repair, which is error free, right? Why it is error free? Because we will utilize the information from the homologous chromosome. See guys, we have the two chromosomes, one from father, one from mother, two copies we have, right? Okay, and both have the similar, we can say these are called as homologous chromosomes. So if there is a damage in the one part of a gene, we can actually take information from the other if it is undamaged, and we can repair it. So why the NHEJ is most commonly? Because guys, most of the time the DNA is highly condensed and it is very difficult to take the information from the homologous chromosome, right? Okay, so to take the information from homologous chromosome, it is very difficult because they are condensed. That's the point. Yeah, so that's why NHEJ is being utilized. This is life-saving to the cell, right? Just join the DNA, otherwise there will be apoptosis because the free ends of the DNA exposed. That will cause initiation of apoptosis, right guys? So that is why the answer of this question is NHEJ. Guys, there was a question asked in the recent time that which phase of cell cycle is most radio resistant, right? There was a question that is most radio resistant phase of cell cycle? So guys, what is the answer to that? Answer is S phase of the cell cycle, or even the G2 can be the answer. Best is go with S phase, because this is a phase during which the DNA is about to replicate and it is uncondensed, right? Okay, so whenever the radiotherapy damages the DNA, okay, it is repaired effectively by homologous HDR, right? Homology directed repair or homologous end joining, right? So that is why, you know, the S phase becomes most radio resistant. Got my point? Why it is most radio resistant? So this is answer will be why it is most radio resistant, due to homology directed repair. Fine. Hope you understood this concept. Yeah. Let's move to our next question. Fine. Eukaryotic genes that produce mRNA. So guys, this was again the recent 1.5 years back question. This was a question asked. So first option, contains the Tata box downstream. No guys, this is upstream, right? Tata box is present at around -25. Fine, in the eukaryotic genes. Eukaryotic genes are bigger. May contain enhancer regions, which is thousands of bases away from the promoters. So guys, that is the correct answer. Are transcribed by RNA polymerase 3? No. The genes which makes the RNA are transcribed by RNA polymerase 2, right? Contains the long stretches of thymine nucleotide that produce a poly A tail. Guys, poly A tail is the post transcriptional modification at the 3' of mRNA, right? Post transcriptional modification. So that cannot be the answer. Genes don't have, you know, what this option is saying is that genes have the T stretch of the thymidine, and when the mRNA is made, it also has adenine. So that is not correct statement. Okay guys, sorry. So hope you understood all this concept. Okay, so that is your eukaryotic genes that produce the mRNA. Now guys, before moving further, first of all, let's look at this point. They are transcribed by RNA polymerase 3, right? Guys, RNA polymerase 1, you know guys, this makes the tRNA. 2, this makes the messenger RNA and the microRNA also, right? MicroRNA also. And 3, RNA polymerase 3, that makes the ribosomal RNA. Just we'll change this, just a second. So this will be making ribosomal RNA, okay, and this will be also making ribosomal RNA, but only one. Fine. So this will be mostly T for 3, that is tRNA plus the 5S ribosomal RNA, right? 5S ribosomal RNA. So there are 3 more types of ribosomal RNAs, that are 28S, 18S, and 5.8S for the Svedberg unit. So guys, don't forget this. This has been already part of these exams, right? RNA polymerase 1, 2, and 3. And guys, there is a toxin which has not been asked till now, alpha amanitin. So guys, this is derived from a mushroom that has a similar name, that is Amanita phalloides. So this is a very deadly toxin which inhibits the RNA polymerase 2, right? RNA polymerase 2 is inhibited by the alpha amanitin. And this toxin is, the mushroom is also called as the death cap-like structure of the mushroom. Death cap. Okay guys, so death cap. After this, let's move to our option B again. So guys, talking about enhancers, let me show you first guys what are the enhancers. Yeah? So this is the gene, right? If I start from, let's say this is promoter, and here we start the gene that is you know, having the first codon in the mRNA will be produced later on, right? Okay, so this is the core promoter, first of all, right? This is the promoter. Then there are some proximal promoter elements which help the promoter activation, like, you know, GC box in the humans, right, for example. Then guys, we have something called as locus control region. Locus control region is again present away from the promoter, and that is not present in all the genes, but in few genes only. That is like the genes for production of hemoglobins, for example, and MHC complex. Guys, LCR is not found in all the genes, but it is found in the genes during, especially genes of embryogenesis, for example. For example, the genes which are producing hemoglobin. So we have the different types of hemoglobin during our growth, and MHC. So they have a particular important region that is called as locus control region. Though never being asked, I'm just telling you. And guys, we have silencer and enhancers. So guys, these silencers and enhancers, they may be present far away, thousands of bases from. So this was a recent question, right? I think it was in the last exams, right? So enhancer and promoter. So guys, they are far away, but they can bind with each other, right, using some proteins. So enhancer binds some proteins, let's say this blue one, and let's say promoter binds a protein, that is black one, and then there is a red one that is called as a mediator between them. So there are various protein complexes which bind with each other and makes a complex like that, because that's how the enhancers are very, very. So this question again has been asked, and nothing new, that is always we read about them, right? So it was asked. The good thing about exams is that most of the time they have questions from my subject repeat. Okay, so that makes it high yield. But the point is how much we revise, because it is little bit, I mean, like you may disagree with me, but it is little bit volatile, so just keep on, right? Okay, so enhancer, guys, now let's write few more points. Enhancer may be located 5' upstream or downstream of the gene it regulates. Okay, fine. So it may be present anywhere, right? Enhancer doesn't need to be present at the site which is close to the promoters, right? Enhancer do not act on promoter itself, that we all know, right? Mediators are present. Enhancer may be found in introns and also in the exons, right? Okay, they may be present anywhere, fine. Even if we reverse it, the change the sequence, it will still be working, fine. And if they are excised and inserted elsewhere in the chromosome, they are still able to do their effect. Okay, so that's how, guys, these are the points on the fine. So which of the following is not, next question, let's start with the next question. Which of the following is not true of post transcriptional modification of mRNA? Post transcriptional modification. So guys, there are three post-transcriptional modificational: 5' capping of 7-methylguanosine, 3' poly A tailing, and splicing. These are the only three processes that are part of post-transcriptional modification. Right, guys? So splicing is mediated by a ribozyme, option A. So guys, that is correct. Question is not true. So what are the ribozymes? That is called as SNRNP or the SNRP proteins. Right, SNRP protein. So guys, can you name me some more ribozymes? So the more ribozymes we can write here, that 28S ribosomal RNA, and guys, this is inhibited by a toxin that's called as ricin. Any other, can you name me? The other is called as RNase P, right? So guys, all part of MCQ, names of ribozymes. Fine, you should know the names of ribozymes. Yes, Avantika, right? You are right. Okay. Then option B, 3' and has a poly A tail. So that is correct. 5' cap engages the critical translation factors to recruit the ribosomes. I will show you again. That is also correct. 5' cap engages the translation factors. And guys, option D says selenocysteine mRNA has post-transcriptionally modified UGA codon. No, that is co-translational first of all, and codon is not modified. It's the tRNA which is modified. So there are two statements which are wrong here. It's not the codon which is modified, and second is not post-transcriptionally but the co-transcriptionally being modified. Getting a point? Okay. So very important question is, yes, you are right. Okay, that is the example of co-translational, okay, and not transcriptional, co-translational modification of the serine tRNA. So the tRNA which is carrying the which amino acid? Serine. That is converted into selenocysteine. Selenocysteine is always important, and especially in the form of proteins which contain selenocysteine like glutathione peroxidase and deiodinase. That you should never forget. Recent MCQs again, right? And deficiency of selenium or selenocysteine, and the same thing, that is called as a Keshan's disease, right? Okay, Keshan's disease. Clear, guys? So let me show you this point, that because during translation, right? During translation, the cap structure is very important because it binds with the eukaryotic elongation factor 4E, right? And the tail binds with the poly A binding protein with the help of G, EIF4G, right? That you should know, the basic thing. Fine. And let me tell you again regarding the selenocysteine mRNA. A selenocysteine mRNA has something called as selenocysteine insertion segment that is present in the 3' UTR untranslated region of the mRNA. 3' UTR. Okay, guys, so there it is present. Fine. And see, guys, this is a UGA which is a stop codon, but this UGA is not stop codon. This is the UGA which codes for selenocysteine, right? So this is a specialty of the mRNA of selenocysteine, never being asked, especially the this point, this point of view, they have never asked like what kind of mRNA it is, right? Okay, so this is a structure which is a stem loop structure, as right? Okay, so guys, that is your question that we have covered, so which is not true, right? Now splicing is mediated by the SNRPs, right? That I told you, which is an example of ribozymes. Question 11. Which of the following is the binding site of microRNA on the target? So what is the binding site of microRNA? His answer is 3' UTR. Fine. It is 3' untranslated region. Okay. 3' untranslated region, right? So not the 5' UTR, not the first codon, not the smallest intron, etc. So guys, what is microRNA? Guys, microRNA is something which is produced from the microRNA genes, right? First, and then it is modified in the cytoplasm also, right? So microRNA was a Nobel Prize in 2006, and the upcoming drugs are based on this concept. Okay, there are drugs are based on this concept, like inclisiran, the drug which is called as inclisiran, which is inhibitor of PCSK9, right, used for hypercholesterolemia. That is based on the concept of microRNA. So guys, what you should know here is that the 3' UTR is the binding site. Okay, fine. See guys, microRNA is produced in the nucleus. Then when the mature microRNA is produced, the mature microRNA is produced using some proteins, right, which are called as DGCR8. So these are present in the nucleus, fine. So these are modifying the microRNA, and then there is a protein that's called as a dicer, okay, dicer endonuclease, which makes multiple microRNAs from a single strand, right, dicer. So these are the proteins involved. Then this mature microRNA binds with the RISC complex in the cytoplasm. So this is, let's say, in the cytoplasm. Fine. RISC complex has a very important component that is called as AGO2. Okay. So once there was a question like components of RISC, RNA induced silencing complex. So what are the components of RISC? AGO2 protein is very important here, right? And dicer also considered as a component of RISC, though it was used previously, but it is also part of the RISC complex. So guys, this was a question, what are the components of RISC complex, right? Now when the microRNA binds, okay, where it binds, I told you that it binds at the 3' UTR. Fine. So suppose this is the microRNA which is 5' to 3'. This is a messenger RNA, this is the messenger RNA. and suppose it binds with a microRNA at the 3' site. So guys, when it binds, as we can see, it is not the complete binding. Okay, so complete binding is not done. So guys, whenever there is not a complete binding, there is translational suppression. But when there is a complete binding, 100% binding, okay, as you can see here, there is a 100% binding. So this leads to mRNA cleavage by the ergonaut 2 AGO2 endonuclease. Clear? So by the AGO2 endonuclease. समझ गए? ठीक है? तो ये सब बेसिक है यार, ये सब पहले भी पूछ चुके हैं, आ सकता है फिर से, डोंट इग्नोर इट. आपको पता ही है INI-CET में योर फर्स्ट सेकंड प्रॉफ वेरी वेरी इंपॉर्टेंट, राइट? Sorry. So hope you understood. If there is no 100% matching, as you can see here, there is not 100% matching, right? These bases are not matching, but in this case there is 100% matching, right? Yes, that is, you should know. So again, I am repeating, microRNA is produced initially by the microRNA genes in the nucleus. They are modified by Drosha and Dicer. Then the microRNA, which is very small in size. What is the size of microRNA? It is just 21 nucleotide long. And what is the process it does? It does a process that's called as gene knockdown. Don't forget this point. Guys, what is the term? Gene knockdown. Fine. So gene knockdown is a term that you should know. Fine. So that's a term that we should know. So guys, what is the difference? Like students ask me sometimes, what is the difference between microRNA and the siRNA? Right. So that we should also know. So there are two terms, microRNA and short interfering RNA, right? Guys, microRNA is produced endogenously. That means inside the cell. This is having an exogenous source, right? Like for example, double-stranded viral RNA, right, gave rise to siRNA. So that means it is not produced in the nucleus. So Drosha is not involved here, right? Second thing, this is the siRNA, which is 100% complementary to the target gene. So it is not, or maybe, not always, right? MicroRNA binds 3' UTR, untranslated region of target messenger RNA. So guys, this can bind anywhere, right? This point can be important. This can bind anywhere. Okay. So guys, normally, you know, the siRNA is a term that was initially given to the short interfering RNA, means like, you know, these are the defense against the viruses. So the viral RNA itself gave rise to this thing that is siRNA, and this siRNA itself goes and bind to the viral genes and degrade them, right? Right. So that is how it is acting against the viruses itself. But microRNA is not acting against our body itself. I mean, like, you know, it is controlling, right? It is controlling our gene expression, microRNAs. You got my point, guys? So that is your gene knockdown. Now guys, when somebody asks you what is the gene knockout and gene knock-in, so knockout and knock-in can be done using CRISPR-Cas9. In the previous question, when we were talking about the CRISPR-Cas9, right, there was a question, right? So CRISPR-Cas9 introduced a double strand break, fine, and if you remove the target gene, that is called as gene knockout, and when you insert a new gene, that is called as gene knock-in. Got my point? Okay, so just remember that the CRISPR-Cas9 can mediate, right? That is called as gene knockout, that is deletion of a target gene. Okay, deletion, and it can mediate gene knock-in, that is insertion of a gene, which is still now very difficult. Otherwise, the gene therapies would have been very much possible, right? So gene knockout, like you want to study a particular animal, like what happens to this animal if you remove a particular gene, right? So you can study that what is the function particular, right? Okay, so that can be done by CRISPR-Cas9, right? Guys, and what does a CRISPR-Cas9 mediates? It mediates something that is called as genome editing. So what is CRISPR-Cas9 actually? It's a genome editing. Edit means insertion deletion editing tool, MCQ, right? Genome editing tool, right guys? Okay. So hope you understood this point. Now moving to our next question, that is let's start with our next question. Incorrect statement for the DNA methylation. So guys, DNA methylation is a process that is called as an epigenetic phenomena, right? Okay, so epigenetics. So in the epigenetics, there is a change in the gene expression without change in the gene sequence. So there are three mechanisms of epigenetics that at least you must know the names, right? What are the names of them? First is called as DNA methylation. Okay, one is called as DNA methylation. Other is called as histone modification, like histone acetylation causes gene activation, right? So histone modification. And guys, the third we have discussed, that is called as RNA interference. Okay, RNA interference, or that what we have discussed is gene knock down, right? Gene knockdown. So that is through the microRNA. So these are the three mechanisms of epigenetics that you must know, right? What are the three names of mechanism? Now coming back to our question, DNA methylation. So guys, X chromosome inactivation, correct. That is through the methylation process. Then associated with gene knockdown, so that is our answer, no, right? Now let's look at the other option. Base excision repair removes the methylated nucleotides. So guys, methylated nucleotides are the wrong nucleotides. So obviously the base excision repair wrong nucleotide, right? For example, when we talk about cytosine, guys, this is cytosine which has NH2 group. Okay, it has NH2 group. Now on this cytosine, if I add a methyl group, fine. So this cytosine has now methylation, fine, and methylated cytosine. So that is called as 5-MC. That is done on the CpG islands. DNA methylation is normally done in the CpG islands. And guys, if there is a removal of this amino group, fine. So this will be converted, suppose if it is also oxidized, so this will be converted into something that is called as thymidine, right? Now in a DNA where there was initially was C which was bound to G, but because of the changes this becomes T and it is still bound to the G. So guys, though it looks like a mismatch, right, but the enzymes will identify that this is not T actually, but it was previously C. So this T has to be removed. So we will be utilizing base excision. Okay. So that is why this statement is correct. So base excision repair removes the methylated. Fine. Okay. Now associated with genomic imprinting, again guys, that means we have certain genes, almost 100 in the numbers, which we get only from one parent in the active. So we are not getting two copies of a particular gene, 100 in numbers. We are getting only one gene which is from the father, let's say, which is active, and sometime something which is we are getting from the mother, which is active. So the other one which is we are getting from father is inactive, right? So mother one is active here, for example. So that is a nature's way to say no to the same sex couples can't have the babies. Why? Because we have one copy of a particular gene, right, from the father and one copy from the mother. Only 100 in numbers, not all the genes, right? So obviously to make a viable embryo, you need both male and female, right? Because certain genes are only given by mother, not by father, and certain genes are given only by father, not by mother. Okay, so that is the genomic imprinting that and it is associated with the splicing regulation. Guys, regarding the option E, that may be new to you. But just remember that splicing regulation, the introns are less methylated than exons, right? So basically there is a methylation difference between introns and exons. That's how it can methylation, to say splicing pattern. And guys, always remember DNA methylation causes gene silencing, right? What does it cause? Gene silencing. Don't forget this, right? During cancers, there is a hypermethylation of CpG islands, right? So methylation is normally done at something that is called as, what is the site of methylation? It is called as CG islands, right? And CG islands are close to promoters most of the time. And guys, promoters are most important for initiation of transcription or making of RNA, right? So that is why if they are methylated, methylation of this cytosine signals gene inactivation. Okay, so in a gene, the area which is rich in C and G, CG, CG, CG is called as CpG islands. And suppose this is the area which is called as promoter. So this C, if it is methylated, this signals inactivation gene. Okay, because those factors which are supposed to bound the P, now they will bind at C. There are various mechanisms regarding that, not out of our course. So let's move further. Fine, guys. So that is your question number 12. Done. Okay, so that is regarding process that is epigenetics. DNA methylation. Clear? Yes. So now, guys, let's start. Question number 13. Lac operon is expressed when? What is the answer? So, guys, lac operon. Operon concepts are only found in eukaryotes, right? What does operon means? Multiple genes under a single. What is the meaning of operon? Operon means simply the multiple genes which are controlled by the single, controlled by the single promoter, right? Single promoter. That's called as operon. So guys, operon is a long process. What we have to remember in operon is that suppose this is a gene, fine, in a bacteria, and in this gene we have a particular area that is called as inducible gene. Then we have a particular area that is called as cyclic AMP cap binding site, which is very close to the P for promoter. And then there is something called as O for operator. And then there are three genes in the lac operon. They are called as ZYA, right? There are just three genes, okay, ZYA. So guys, these are the three genes which make the mRNA, then it makes the proteins or the enzyme for the lactose digestion. Right, now the point is bacteria doesn't use, normally it will use glucose. So guys, unless until glucose is available, lac operon is not expressed, right? Okay, so when the glucose is present, so lac operon is not expressed. Fine. Now guys, when the glucose is not present, it is starvation and cyclic AMP levels increase. So in the absence of glucose, there is increase in cyclic AMP, and this cyclic AMP binds to the cap, that is called as catabolite activity protein. Fine. So in the absence of glucose, so this cyclic AMP and cap binds here, fine, and it helps the RNA polymerase efficiently transcribe. So this is the RNA polymerase, suppose, and this is cyclic AMP and cap, right? Okay, second. So that is your promoter. So promoter normally binds a RNA polymerase, and this is helped by the cyclic AMP cap complex. So guys, hope you understood this point, right? The only possibility of the expression of lac operon is glucose must be absent and lactose must be. Okay, so glucose must be absent and lactose must be present, because whenever there is a glucose is absent, guys, this glucose will this glucose will cause the increase in cyclic AMP, right? Increase in the cyclic AMP. Fine. And guys, lactose is something which is inactivator of repressor protein, right? Because this is the gene which makes a repressor protein, and this repressor goes and bind here at the operator and prevents the RNA polymerase to move. That is all about the lack of, right? Okay. So again, cyclic AMP is something which activates. Lactose is something which helps in activation by removal of lactose by repressor. Hope you are done with this, right? So that's all about the lac operon. Moving to the next point, that is again related with the regulation of gene expression, which is a very important topic. Regulation of gene expression. Fine. So in the iron deficiency anemia, right? So guys, in the iron deficiency anemia, there are two genes, ferritin and transferrin. The ferritin gene, which is required for storage of the protein, is inactivated, obviously, because iron is already deficient. And transferrin, so in this question we have transferrin receptor based on the transferrin receptors. Okay. So in the presence of iron and in the absence of iron, what happens? Let's see this using this simple image. Okay. So guys, this is the one which is talking about ferritin. Okay. So when there is no iron, there is a protein known as iron regulatory protein, as you can see here. Okay, iron regulatory protein. So this iron regulatory protein binds the mRNA 5' UTR. Okay, 5' UTR of the mRNA. And guys, because of this binding, the ferritin mRNA cannot be translated, as it is written impeded, right? It cannot be translated, right, in the absence of iron. Now in the presence of iron, the iron regulatory protein will bind iron, so the mRNA of ferritin will be free to get translated into protein. Got my point? So IRP is iron regulatory protein which binds with, but if there is no iron, then IRP is binding to the mRNA of ferritin and prevents its translation. Now looking at the transferrin. Now guys, in the transferrin, first of all, remember that 3' UTR has some stem loops like as shown in the figure. So if there is no iron, the IRP is again not bound to the iron, so it will bind at these stem loop sequences which are called as also IRE, iron regulatory elements, right? Okay guys, what do you call these? I can name them as iron regulatory elements, right? And this is IRP. So when they are bound to it, there is mRNA stability, and guys, there is increase in the translation of transferrin receptors. Guys, obviously, first of all, think like this: iron is deficient. I don't want, so its mRNA should be stopped. I want transferrin because it should be ready to take any of the iron which is, so transferrin mRNA should get translated. So during this process, which is given in our basic books, has been asked in exams 2 times, right? What we should know is that this point is given as the answer. See, iron, in the iron deficiency anemia, iron does not bind to the IRP, right? Iron regulatory protein, which results in translation of transferrin. Very simple. Now look at the other option. You rule out. Iron does not bind to iron regulatory protein and doesn't cause translation. That is wrong. Iron bind to iron regulatory protein. So there is no iron first of all. So this option is totally nonsense, right? Iron is not at all available. Iron binds, again, there is no iron यार, कहाँ से bind हो जाएगा, right? तो ये 2 ऑप्शन तो वैसे ही रूल आउट हो गए क्योंकि आयरन डेफिशिएंट है. तो यही 2 ही ऑप्शन है हमारे पास और यहाँ पे ऑप्शन बी करेक्ट सर. Get my point? Fine. So that is what happens in the iron deficiency anemia. Clear to all? Okay. So those who will be watching later on, right, I hope you'll be able to understand this very easily. Okay. So iron is not there. IRP binds at the 3' UTR of transferrin receptor. Don't forget this 3' UTR and 5' UTR. Because in ferritin mRNA we have 5'. See, if F for ferritin, F for 5. Right? T for transferrin, T for 3. Right? So 3' UTR has a IRE. Iron regulatory elements are present in 3' UTR. Okay, even in selenocysteine, the 3' UTR has a stem loop sequences. I told you. Even the microRNA binds at the 3' UTR. So 3' UTR has a very much importance in this section, right, guys? Okay. So that is all about this point, right? And guys, when the IRP in the presence of iron, the IRP is something which will bind with, let's say, iron. So they will not bind at the mRNA, and this will lead to degradation of mRNA of transferrin mRNA. Clear? Let's move to the next question. Incorrect of mitochondrial DNA. See guys, mitochondrial DNA is a very, very important topic, favorite of examiners. Okay. So first point, higher chances of mutations. Yes, correct. Why? Why there are higher chances of mutations? Because of the presence of electron transport chain in the mitochondria, and the DNA in the mitochondria is lying free. It is not protected by any separately. So there are high chances. And second thing is, most of the DNA in mitochondria is coding, right? So there are no introns, or there is no junk DNA, right? So there are higher chances that the X-rays will always affect the coding region, which is almost 97 to 99%. Okay. Double stranded circular, yes, right? Mitochondrial codons are different from nuclear codons. Yes, guys. For example, AUA is methionine, UGA is tryptophan, and AGA and AGG are stop codons, right? So they are stop codons. Clear? Option D, that is our answer. Mitochondrial DNA codes for all the ETC complexes. No, guys, not the complex number 2, right? Not the complex number 2. So complex number 2, succinate dehydrogenase, it is made by the nuclear genes and imported in the mitochondria, right? Okay. Okay. So what else we need to know about mitochondrial DNA? Guys, mitochondrial DNA has written double stranded DNA, and it is circular 16569 base pair compared to 3 billion in the nucleus, right? 97% is coding here, right? There is only one polymerase gamma which has all the activities, that is proofreading and polymerization, right? Polymerase gamma is found, okay, in the mitochondria. Then there are 37 genes are there. Out of these 37 genes, 22 are tRNA genes. Only 2 makes the ribosomal RNA, and 13 are the protein coding genes which makes the mRNA. Proteins, I told you, ETC complexes are made except the complex number 2, right? 37 genes. These genes are contiguous, right? That means there is little separation of 1 or 2 nucleotides. So genes are contiguous, and there are some genes of ATP synthase which also overlaps, right? Fine. So that is also one of the point. Then the mitochondrial DNA follows a principle of heteroplasmy, means the multiple genotypes are present in the same mitochondria. There are 10 copies, and the 10 copies may have the different genetic sequences, right? So that is called as heteroplasmy. And multiple copies, so that's called as polyploidy, right? We all know that there is a maternal inheritance, okay, maternal inheritance, okay. And one more point is that not all proteins, okay, not all proteins are coded by the mitochondrial genes. I told you complex 2 by the, right? For example, complex 2 of ETC is coded by the mitochondrial nuclear genes, right, guys? So these are the points that we should know. And what are the names of mitochondrial disorders? Guys, there are some names which you must know, like Leber's hereditary optic neuropathy, and in this case there is a complex 1 disease, and mitochondrial encephalopathy, lactic acidosis, and stroke. Guys, that's a complex 4 disease. 4 and 1 actually both. Okay, etc complex 1 and 4th. Clear? So that is your mitochondrial disorders. Fine. That's all about the mitochondrial genes that you should know. Fine. So that's a very important question. Question number 15. So guys, moving on to the question number 16. The sigma subunit of RNA polymerase. Guys, that's a very important enzyme that's called as sigma subunit. Right. So this sigma subunit of RNA polymerase identifies and binds the -10 promoter. Fine guys, -10 promoter, that's called as a Pribno box, the bacteria. Okay, so this is the most important subunit, right? So this sigma, we can say this is most important for the transcription initiation, right? Transcription initiation. Fine, that is why. Then it helps in termination of transcription. No, guys, that is the rho factor. This is a symbol, rho factor. So this can be important. Sigma has been asked previously. Inhibited by rifampicin is the beta subunit. Guys, this one. Fine. Required for proofreading activity of RNA polymerase. Guys, RNA polymerase compared to DNA polymerase. Remember that RNA polymerase, when we compare it from DNA polymerase, no primer is required, no primer needed, and there is no proofreading activity is there, right? So that's the difference of RNA polymerase and DNA. So guys, if there is no proofreading, what is the direction of proofreading? 3' to 5' exonuclease. So there is no activity of RNA polymerase in 3 to 5 direction, but that is found in DNA polymerase. Because this can be important. That means RNA polymerase is only working in 5 to 3, never in 3 to 5, right? Okay, so that is all about the RNA polymerase of the bacteria. I told you already about eukaryotic RNA polymerase, right? Following property of genetic code is explained by the wobble hypothesis. So guys, wobble hypothesis means imprecise pairing. Wobble means what? Imprecise pairing. Okay, and that one amino acid can be coded by multiple codons. So what is wobble hypothesis means? That the multiple codons, right, can code for single or the one amino acids. Okay, multiple codons code for the single amino acid. That is called as wobble hypothesis. So what do you call this process? This is called as the degeneracy, or it is also called as redundancy, right? As degeneracy means what? Like, just to say one sentence you have multiple languages, right? Just to say I love you, you have 300 languages, right, in India. So that is called as degeneracy. So just to make, let's say, arginine, you have 6 codons, for example. Similarly, right? So that is option B, guys, degeneracy, right? Fine. So universality, and these are the other features of the genetic code, right? Let me explain about the wobble hypothesis, guys, with this figure. For example, these are the 2 codons which codes for leucine, CUC and CUU, right? So where is the wobbling happening? Wobbling means imprecise base pairing. As per the Watson and Crick, A should bind with the T and G should bind with the C. That is called as Watson and Crick base pairing, right? But later on it was found that we have multiple codons for, we have 64 codons for just, let's say, 20 amino acids, right? So this is due to the imprecise base pairing at, see guys, wobbling occurs at, first of all, you should write this line, that wobble base pairing, where does it occur? Right, it occur at the 3' of the tRNA anticodon and the 5', right, or the first base of, that is it already. So 3' of mRNA codon, corrected, mRNA codon as we will see in the figure, and 5' of tRNA, okay, tRNA anticodon. Okay, समझ गया? So let's see this again. So this is the 5' of tRNA and 3' of the mRNA. See, normal base pairing is G and C, but G can also bind with U. So that is wobble base pairing, right? G can bind with U, is wobble, that is imprecise, that is not strictly followed. Okay, but here it is done. So that is called as wobble. Right, guys? Okay. So there are various examples of wobble base pairing, guys. Even the tRNA, may contains inosine, and I, can also pair with U or the C or the A in the messenger RNA 3'. Right? So the third base of the messenger RNA can also base pair with inosine, right? If it is U, C, and A. Like there are a lot of examples. You don't have to remember that. You just remember this concept that wobbling explains degeneracy of, right? And where does it occur? Location you should know, right? That's something basic. Fine, guys. Okay, moving to the next question. Identify the following initiation complex. So guys, previously there were a lot of questions on these initiation complexes in the like PJ and all. Sorry, and even in AIMS. So they have not been repeated in the last 3-4 years, so can be repeated, right? I feel like that. So I have put this question: identify the following initiation complex of translation. Obviously we are talking about translation initiation complex, right? So guys, if we look at the complex here, we have messenger RNA present here, right? Then we have 40th subunit of ribosome present here. Okay, so mRNA plus ribosome, and so it is not shown here, right? The tRNA is not like shown here clearly, but there is also the tRNA, fine, of methionine. So this is the 48S complex. So guys, in the 43S complex there is no messenger RNA. And why it is not 80S complex, right? Because 80S complex has 60S subunit, which is not shown in the picture, right? Simplifying it again. So initially what happens, the 40S binds with the tRNA with methionine and some factors. So that is called as 43S complex. Then mRNA binds, that's called as 48S complex. Then the larger subunit of ribosome also binds, that is called as 680S complex, right? So that is your initiation complex. Clear guys? चलो आगे चलता है, ठीक है? तो यार ये सब पढ़ लेना, क्योंकि देखो नहीं पूछा कुछ टाइम से, बट वी कांट इग्नोर, है ना? ऐसा हम कुछ इग्नोर नहीं कर सकते इसको, राइट? क्योंकि पूछा हुआ है पहले. दे हैव आस्क्ड दीज क्वेश्चन प्रीवियसली, not as the image based but component based. Sorry. Right guys, now I'm just taking one point that is called as a recombinant DNA, which is considered as very important and seems to be little difficult, but it is not, right? So in the recombinant DNA technology, first of all, the simple meaning, what is a recombinant DNA? That is also called as the chimeric DNA, right? Chimera means like the fusion of two animals, like the body of horse with the face of lion. Okay, so that is chimera. So chimeric DNA. So guys, that is also called as recombinant DNA. That means the fusion of, let's say, bacterial and human DNA or plasmid and human DNA. That is called as a. So first of all, what we need is to make a recombinant DNA, we should cut the DNA using a restriction enzyme. Because restriction enzyme, they cut at, as you all know, the palindromic sequence. That is just 4 to 7 base pair. Like for example, GAATTC. So what is a palindrome? Again, it will be GAATTC. Okay, in a double strand DNA. So if I read from 5 to 3 and again 5 to 3, it is same. That is called as restriction enzyme, right? Because restriction enzymes are normally found in bacteria and their defense against the viruses like phage, okay, bacteriophage. And how does a bacteria prevents its own degradation by the restriction enzyme? By methylation process, right? So if the bacteria methylate its adenine at the site which is called as restriction site, the self cut will be avoided. MCQ, right? How does the bacteria avoid the self cut of the DNA? By putting methyl group. It has a lot of importance. By putting the, okay, and they are named like this, like for example eco R1, derived from E. coli. So that's it. So we took the DNA, cut at the restriction sites, make thousands of fragments, right? And guys, these fragments may be sticky and they may have the unpaired basis. Then next point is cloning. Guys, what do you mean by cloning? Cloning means replicate. Okay, we can say replicate or propagate. Fine. Okay, to make copies, right? To make copies. Because before the PCR was invented, fine, we had this process only to make the copies of DNA, right? Still it is used, though it is laborious process, but it is a safer process because in the PCR we use a lot of heat. So what is cloning? Suppose I take the human insulin gene and add to the plasmid, right? That is called as a recombinant DNA. And then I transfer this recombinant DNA to the E. coli where the plasmid grows. And when the plasmid divides, I get multiple copies of insulin. That is called as cloning process, right? So previously, DNA library was a very important term. Why DNA library means? DNA library means simply the stock, right? Stock of, let's say the known gene fragments, right? Known gene copies. Okay. Suppose I want to study a gene, right, of a particular organism, and that is found in oceans, right? But right now I don't have that access to the ocean. I'm sitting in a landlocked. So what I can do is I can ask for the gene. Libraries are nothing, just talks of particular gene. So don't worry, nobody will ask you the meaning of DNA, right? But they will ask you the question that what are the two types of DNA libraries? Okay, so guys, there are two types of DNA libraries. What is DNA library? Stock of the genes or the DNAs. So guys, genomic library means, whole organisms DNA, right? Whole organism DNA, okay, which include the introns, means non-coding, and also some areas like promoters. What is cDNA library? So only the coding genes, right, which make mRNA or the proteins. Fine. So what we will be doing, we will convert the mRNA to the DNA. That is called as complement, cDNA. So in this case, we are using only the coding genes, right? So it has no introns. That means it will not have any introns. Only the coding sequences will be available. So if I want to study, let's say the insulin gene, so that will be from a cDNA library I can take, or genomic library, I can also take from that, right? But genomic libraries are not prepared for the complex organisms or like a huge DNA, right? So for the huge DNAs like humans, we prepare cDNA library. Nowadays, they're not commonly used because of invention of something. Whatever the gene I want, I can, actually using the primers, I can amplify that sample, right? You got my point, guys? But you should know the definitions of cDNA and G. I mean to say the differences. Next point. So, guys, during making of a recombinant DNA, we take the restriction enzyme. We cut the vector's DNA, that is, let's say plasmid, and human DNA with the same enzyme like EcoR1. So they will have a sticky ends, and so they will fuse easily. So that's called as a recombinant plasmid. Okay. And now I can propagate this recombinant plasmid to the E. coli and get the multiple copies of this dead gene. Got my point? Okay. So that is how the recombinant DNA is produced. Now guys, talking about the vectors, right? Vector means that carrier of DNA, right? So vector means carrier of DNA. So DNA carrying capacity of vectors, like plasmid is the most common vector. So it can take only 10 kilobase, and this is the most common vector in genetics, MCQ, right? Because of its properties that I will tell you. Phage can carry around 20 kilobase, cosmid can carry 50 kilobases, bacterial artificial chromosome up to 300 kilobases, and yeast artificial chromosome up to 1000 kilobases, right? Okay, so DNA carrying capacity of vectors. Now guys, what are the properties of vectors? Vectors should have properties. Okay, so what vector should have? Vector should have one origin of replication, right? Then it should have some restriction sites, restriction endonuclease sites, so that it can be cut easily, right? Then vector should have, a marker, selectable marker, right? Selectable marker, like for example tetracycline resistance gene, for example, yeah, ampicillin resistance gene, right? Why you want that? So that you can select the colonies of the bacteria which contains the plasmid which has ampicillin or tetracycline resistance gene. Okay, because it will grow in the medium which contains tetracycline, right? So you can identify ki, okay, this is the bacteria which has this particular copy of plasmid. Got my point? Fine. Now guys, vectors, these are the properties to select a vector, right? MCQ. Now there are two types of vectors, expression vector and cloning vector. Now guys, the figure that we have seen, okay, the figure that we have seen here, that is called as a cloning vector. Okay, so what is the difference from expression vector? So guys, expression vector has expression. First of all, understand the expression vector. Expression vector is something we used to synthesize the human protein in the host cell. Let's say, for example, if you want to synthesize insulin, humulin, right? So you need expression vector. It will express. So how will you differentiate in MCQ the cloning vector and expression vector, right? How to differentiate that? So guys, obviously I want the vector to produce the protein. So what we have to do is we have to add a promoter, right? Promoter. What is the importance of promoter? Promoter binds the RNA polymer bacteria. Suppose this is a plasmid and this green color is the DNA insert or human gene. Fine. Okay. So why the bacteria will make human insulin? It will not make it until some stage. So what we can do is to make a plasmid, to make plasmid able to generate insulin, what we have to do is we have to add a promoter which is identified by the bacterial RNA polymerase, right? And we have to add certain sequences like Shine-Delgarno sequence, right? Shine-Delgarno sequence is a sequence which is the ribosomal attachment. Okay, so once the mRNA is made, it has this sequence which can bind the ribosomes and the protein, right? So what are the points again? So if we compare, so expression vector, right, compared from the cloning vector. Guys, what is the difference? The expression vector compared from cloning vector has a very strong and easily inducible promoter, right? So which can be identified by the host cell, host RNA polymerase, okay, to make some proteins, and it has the Shine-Dalgarno sequences or the Kozak consensus sequences, which are ribosomal binding site. Okay, ribosomal, because that will be expressed on mRNA, na? Ribosomal binding sites. Obviously, their SDS is on the gene, but it will appear on the mRNA. Clear? So that is called as expression vector. Okay, guys, nothing much. Don't look much on this figure. I meant to say I have explained the basic points. Okay, so cloning vector will not make any protein in the host cell, but expression vector will make protein. Now you understand that how we synthesize the human proteins, that I will tell you again. Okay, how to transfer the recombinant DNA to the host cell, right? So guys, the most common plasmid that we have modified. So now we have a plasmid which has added human gene plus promoter plus the SDS sequences. Got it now? So there are some methods: electroporation, microinjection, and gene gun. So these are some names. And incubating the cells with calcium. So guys, these are again old MCQs, can be repeated, right? Recombinant DNA technology is always important. This question also has been asked in AIMS. So guys, how will you identify that which E. coli or bacteria has the recombinant plasmid? Now we are sending the plasmid to E. coli, right? Fine, that is like what you call as transformation. Fine. So blue-white screening, guys, as you can see, this is the colony of the bacteria which has been given a plasmid, right, transformed, and the white ones are the one which are successfully having the plasmid which contains the ampicillin resistance gene, right, apart from the target gene, insulin gene. And the blue ones are the failed cases. The E. coli was unable to get transformed. So guys, we will select these white colonies and we will utilize it in the further study. Got my point? So guys, remember this line: blue-white screening. White is success. Okay, here. I wish you the success in the upcoming exam. And guys, again a question: what is the application of the recombinant DNA technology? So production of the recombinant proteins like humulin, tissue plasminogen activator, erythropoietin, factor 8, growth hormone, etc. Right? Then question, guys, again a question: why the mRNA is used in recombinant DNA technology? Guys, because there are no introns. I mean to say mature mRNA. We will use a mature mRNA from the cytoplasm. So there are no introns. And second thing, splicing is absent in bacteria, right? The splicing is absent. So suppose if you give the gene, if you add the human insulin gene, the mRNA will contain introns and bacteria cannot splice them. Can't get the, right? So you have to give cDNA, right? From the mRNA will make the cDNA, and that will having no introns, and that will make the mature protein. Right, guys? What is the most important step? MCQ. Most important step is to add the promoters. Guys, obviously, no. Without promoters, how the RNA polymerase of E. coli will bind to the plasmid gene? Right? E. coli is not bothered if the plasmid is present. Right? Give some stimulus to them that, hey, coli, bro, make some. Right? That is, we have to add the promoters. Right? To fool them. Okay? We have to trick them. Right? So that is application. Guys, second application is site-directed mutagenesis. So guys, what is site-directed mutagenesis? We can alter the genetic codes, right, to make a distant protein. So guys, we can study the pathogenesis of certain diseases. Okay, so we can alter and make the models, like where there are point mutations, etc. Like in a disease, there is a point mutation, right, of one nucleotide change. So that is called as a site-directed mutagenesis. And guys, there are some insulin like Lispro. Okay, Lispro insulin. What is Lispro insulin? Lispro insulin is an example of the site-directed mutagenesis. How? Normally in the insulin B chain, guys, the proline is present first and then the lysine, but it has been reversed and it becomes lispro. Previously it was proline and lysine normally. So lispro, which is better absorption, right? So next is gene therapies. Guys, we can make the viral vectors like adenoviruses, which contains the target gene, which can make the target protein, the human cells, right? So though not very successful, and guys, we can construct the DNA. Fine. So these are the uses. Okay, so this was the MCQ, the INI-CET itself. What is the correct order of isolation of desired proteins via the recombinant DNA technology? What is the answer? Simple. His answer is A. Why? Because protein expression done. Then I will break the cell. Then after breaking the cell, we will do the SDS page. Guys, SDS page is the name of electrophoresis. And SDS page is, only dependent on molecular weight. That is again MCQ. Need PG many times asked. SDS page is a type of electrophoresis, right? Which is based on molecular weight, not on charge. Right? Not on charge. So lastly, we will do the elution. Elution means extraction of the protein from the gel of electrophoresis. Right, guys? So option A is the answer. Clear? So hope you are getting all the points which have been asked in exams and in the very concise, crisp and desired details, right? पढ़ेंगे उतना ही जितना ज़रूरी है, right? We are not going into unnecessary details. These are all the points which have been part of MCQs or have been asked in exams, right? Okay. Techniques to study, guys. One more thing, when I was talking about this SDS page, right? So it is dependent on molecular weight. Now, guys, to separate the DNA, this is for the protein actually. SDS page is protein separation. To separate the DNA, we have electrophoresis that is called as PAGE, polyacrylamide gel electrophoresis, right? So that is one of the best type of electrophoresis to identify the DNA with the small differences, right? High resolution means the DNA which is differing in the few base pairs only be identified, right, by the PAGE, polyacrylamide gel electrophoresis. So that has a very high resolution for DNA separation. Okay, the DNA separation, right, with small differences. Small difference in size. Okay, might not be able to write it here. I have written for DNA separation with the small differences. You might not be able to see because of my image, right? Okay, let's move to the next topic. Techniques to study the DNA protein interaction. So guys, that is called as DNA footprinting, right? Footprinting, DNA protein interaction. Fine. Okay, so guys, northern blot is to analysis of gene expression, right? So guys, what do you mean by gene expression? Gene expression means either the RNA is formed or the protein is formed. Fine. So for the protein, okay, so for the protein and RNA, we have the different techniques. So the protein, if you want to identify, we have Western blot, right? Western blot. RNA, if you want to identify, we have northern blot. Then we have RT-PCR, okay, reverse transcription PCR. Fine. Then we can also take micro arrays. I will talk about this in the last. Micro arrays. Okay. And there is one more name that is called as RNase protection assay. Fine, guys. So these are the names like which can identify RNA or which can actually identify the gene expression. So even the Western blot is correct. If the question is which of the following technique we can use for identification of the gene expression, so these all can be the correct answer, right, guys? Okay. Let's move to the next point. Fingerprinting, guys. I just want to tell you one thing here. DNA fingerprinting is based on the variable number of tandem repeats and short tandem repeats, right? So if this question is asked in exams, just mark based on short tandem repeats the better answer than the variable number of tandem repeats. Okay, short tandem repeats, right? Like microsatellites, 2 to 6 base pairs. So that is a better because these are easy to analyze. Okay, fine. Like CDFD in Hyderabad, the PFs, right? Okay, for forensic analysis, forensic criminology. So where the fingerprinting is done, so that is based on STR commonly, right? Okay, guys, this was the question. COVID can be diagnosed by all of the following except. So the answer for this question is C, that is called as southern blot. You know, guys, why? First of all, COVID, we can't do southern blot because southern blot is, you know, if we look at the southern blot need electrophoresis. I'll tell you the steps later on. And guys, we don't have that much of sample during COVID analysis, like you're taking the sample from the mucosa, nasal mucosa, you know, the sample amount is very low, right? So what you have to do is first convert RNA to DNA and then you do the PCR, right? In southern blot, you have to do the electrophoresis. For this, you need a high quantity of, so for that you have to do PCR before the southern blot, no? So why to do southern blot at all, right? And southern blot is for DNA, simple. Even if you know nothing, southern blot is done for identification. Though I can do it by converting this into cDNA, but obviously this is a long process. Southern blot is the correct answer, right, guys? Okay. Question number 22, detection of mutation with the low allele frequency. Again, the INI-CET MCQ can be done by, guys, any point mutation, right? Nowadays, the best answer is next generation sequencing. It is also called as massively parallel sequencing, right? Massively parallel sequencing. Clear? Okay, no further details are required as such, right? So that is next generation sequencing. Don't forget Sanger sequencing. That was, I told you, dideoxynucleotide based method. Forget these two options, not at all required. Okay, unnecessary, because I know the answer. Nowadays, the best answer for any mutation analysis, unless until it is not aneuploidy or the you know, the number of chromosomes are increased like trisomy 21. I will go with next generation. That is the best one, especially the point mutation analysis and low allele frequency. Okay, rare type of mutations analyzed by sequencing whole, right? Also called as massively parallel. Yes, next generation sequencing has 3 steps. Again, just like making the DNA library, then doing the PCR amplification, okay, and finally sequencing. Though it was a question in PJ long back, 5-6 years back. Okay, that is like, you know, next generation sequencing. Now, guys, just I have you know, because the time is less, and so I have just compiled some more important points, right, which may be part of MCQ exams or which have been already asked in exam. Let me just read them. Otherwise, we are done. So techniques, just 1 or 2 page. So first point is the quantification of DNA methylation, okay, or the genomic imprinting analysis. Guys, we need something called as bisulfite sequencing. Just name DNA methylation or old question. Detection of histone modification. Guys, we can do mass spectrometry, that is important for analysis of inborn errors of metabolism like maple syrup urine disease. But we can also do it for histone modification analysis and also the chromatin immunoprecipitation assay, right? Chromatin immunoprecipitation assay, right? DNA footprinting, guys, I told you already, right? DNA fingerprinting, I told you that STR is superior, right? Why? Because of their ease of accessibility. Yes, there is one more sequencing apart from Sanger's and next generation sequencing, that's called as pyro. Pyro means heat or the light. Okay, so guys, that is just simply based on the emission of the light signals. Fine. Each time a nucleotide is added, and as nucleotide we take ATP, but what we add is AMP. So the two phosphates will be removed, which are called as pyrophosphates. So that is why it is called as a pyrosequencing. Fine. So detection of pyrophosphate, and that is through the light signals. Microarray, guys, the principle of microarray is DNA hybridization. It's a kind of a chip with known sequence like what is present here, right? So these are the small DNA fragments which are present on the chip, right? So what we can do with the help of microarrays, we can compare the gene expressions of the two cells, right? So we can use something that's called as comparative gene expression analysis, right? Let's say the tumor cell versus the healthy cell, right? Tumor cell versus the healthy cell. Fine, guys. So that is hybridization between two DNA strands is the primary principle of microarray. Okay, so what is the sequence present here? Suppose there is a gene of telomerase, and I just want to know whether the gene of telomerase is present in tumor cell. So this may give me a green signal. But if I take a normal cell, the gene for telomerase, which is not expressed, this may give me the red signal. And if both the tumor and normal cell have the same gene expression, this will give me the yellow signal. Okay, so at least remember this picture. That is a micro or the gene chip. This is also called as gene chip or the DNA chip. Here, PCR steps have been asked. I told you, right? So denaturation, annealing, and chain extension, annealing the primer. Guys, once a question was asked like Taq polymerase has no proofreading. So which polymerase can be used? So guys, the Pyrococcus bacteria gave us something called as PFU, Pyrococcus furaceus. So this PFU or PWO, that's a polymerase which has the proofreading function. Okay, can be repeated again, old question. Blue white screening, I told you. Guys, there is one more type of electrophoresis that's called as pulse field gel electrophoresis. Pulse means, we will change the direction. Normally, let's say this is cathode and anode, right? But we will keep on changing it. So we will making the pulses, right? We will making the pulses like this. So we will keep on changing the electric field. So guys, this is very important for separation of very large molecules of DNA, right? So if there are large molecules of DNA, we need the pulse field gel electrophoresis. Right now it is used in microbiology for identification of bacterial, right? Okay. So this was once a question in DNB exam, but just I have included this name of technique for DNA separation. Page, I told you guys, it has a high resolving power, right? Southern blot, if steps are asked, guys, what is southern blot? Southern blot means to put the DNA fragments on a membrane and then putting a probe which is complementary, right? Suppose this is the DNA, right? I want to identify, right? There are various such DNAs are present, fine, and then I put a probe which will identify the DNA of particular sequence that I want to find, that gene, if it is present or not. It will not bind here, not bind here, bind here, right? But it will bind only at particular point. That is called as southern blotting. It has like, first we'll break the DNA, then do the gel electrophoresis, then transfer the gel to or DNA to the nitrocellulose or negatively charged membranes. Then I will just add a probe which has a fluorescent labeled color like SYBR green. Okay, then we will just wash and detect. Okay, because this has a fluorescent color, it will glow out. That is something that is electrophoresis. Okay, so like that, southern blot. Okay, so what is the importance of southern blot? To identify the gene of interest. Then guys, there is a technique known as fluorescence resonance energy transfer, right? Okay, so this is required for studying the protein folding or the protein. Fine. Okay, there is one more technique which can study the protein folding, especially the secondary structures, circular dichroism. right? And to identify the 3D structure of protein, not the protein folding, but the actual structure of the protein, we use nuclear magnetic resonance and X-ray crystallography, which was also used by Watson and Crick for DNA structure, right? For the DNA structure. Okay, guys. Then we are starting with the F for FRAT, F for FRAP, fluorescence recovery after photo bleaching. So guys, this is giving us the mobility of the molecules in the living cell. Like, you know how the molecules are moving, let's say from the cytoplasm to the mitochondria, for example. So that is, we can do the study using fluorescence recovery after bleaching. That's how we came to know that how does the, let's say, a particular molecule like pyruvate, suppose, moves to the, let's say, mitochondria, for example. How does the citrate comes out from mitochondria to the cytoplasm for fatty acid synthesis, let's say. That is using FRAP, fluorescence recovery after photo bleaching. Fine. Then guys, fish you might also read in your pathology, but just to tell you that the fish is fluorescent in situ hybridization, right? And it detects the deletion, translocation, aneuploidy, most importantly, like trisomy 21 or Down syndrome. Fine. Okay, so this will fluorescently label the, let's say, chromosome 21 with a typical color, let's say like red, for example. Suppose this green color is some other uh, chromosome, right? Okay, so that is an extra copy, na? Suppose this is a chromosome number 1, which is obviously 2 copies only, but 21 is 3 copies. That's how this figure has been asked, like, which is simple to identify. Okay, fish. Then guys, chip on chip is nothing but chromatin immunoprecipitation plus microarray. Fine. So this is similar to DNA footprinting, but this is done in the living cell for DNA binding site of a protein. DNA protein interaction can study using chip on in a living cell. But DNA footprinting I have to do in lab with the help of electrophoresis. So footprinting and chip on chip are similar in this case. As and the last one is RFLP, restriction fragment length polymorphism. So guys, restriction fragment length polymorphism is something which is utilized technique for genetic mapping, right? Paternity test and diagnosis of disorders like sickle cell disease and cystic fibrosis. So guys, this is actually, okay, just ignore them. We have already covered them, right? So this RFLP, right? So how does the RFLP arises? See the name, restriction fragment length polymorphism. Okay, so human beings, they have, you know, differences in the length of the DNA if it is cut with a single, you know, enzyme, right? So how does this RFLP arises? So this RFLP arise due to either the single nucleotide polymorphism, like in sickle cell disease, what happens? You know, GAG, the codon for glutamine converts into GUG on the mRNA. So single nucleotide is changed. That's a mutation. Or it can arise due to the VNTR, variable number of tandem repeats. You know, the length of VNTR differentiators. That is also the basis of DNA fingerprinting, STR or VNTR. Right, guys? Okay. So let's say, suppose this is, just to understand, suppose this is a hemoglobin A, you know, which is cut at these 3 sites, and this is hemoglobin S, which is cut only at 2 sites. Why? Because this site, there is the change in a single nucleotide, which will not allow the restriction enzyme to cut here. You know, it is GAG to GUG. So this site is lost, loss of restriction site. Okay. So guys, after cutting this DNA with enzyme, if you do the electrophoresis, you can actually identify the patient, the heterozygote and homozygous sickle cell patient. Okay. Like, you know, the normal person will give me 7 and 3 kilo base fragment on the electrophoresis, right? And the patient of sickle cell disease will give me 10 kilo base, right? This one, a complete fragment. And guys, if it is a heterozygous condition, okay, carrier, so it will give me all the fragments, because the one chromosome has, you know, the restriction site is not lost, but on the other chromosome or other gene, it is lost on the beta globin gene, right? So it will give me all the 3 fragments, right? 10 also, 7 also, 3 kb also. So this is heterozygous condition. basically. So guys, this is how we can identify disorders, or we can use this in paternity testing, right? Like, the babies have the DNA 50% from mother, 50% from father. So you can actually identify the real parents of a child, because that has the similarity 50% with mother and father, right? 50-50. Clear? So that is fine, guys. So I have tried to cover everything in this lecture, right? Which is part of molecular biology, which is very, very high yield, right? So all the PYQs and all the points which have been part of the MCQ, even in the 5 years or 6 years old papers I have covered, right? Because nothing is old, nothing is new. Though right now you are supposed to do only the 3 or 4 years old PYQs, but still I have included few questions, right? As I told you, like the 43 years complex and all. They have not recently been asked, but can be asked anytime, because they are part of our books यार. नहीं कभी भी आ सकते हैं. तो पॉइंट ये यार कि right now what I can say is all the best, right? Okay. And in the last moment, don't lose the hope. Stick to your plan, right? And keep a healthy diet, which is very important for a right now for your energy levels. So keep eating a very, very, don't miss on that, right? Okay. So guys, thank you so much, right? And all the best. And remember the points I told you. Okay, don't be aggressive while marking the MCQs. Don't take all the 200 MCQs exam. So fine. Thank you so much. And all the best. Bye. And guys, just one more thing I want to tell you.